Hello everybody, welcome to another episode of Biology with Mr. Emond. Today we're going to be looking at cyclins, cyclin-dependent kinases, and how to calculate the mitotic index. So let's get started here. What we're going to be discussing first is basically what a cyclin is. So a cyclin is something in the family of regulatory proteins. So they're going to be regulating the cell cycle right now. So they're controlling the progression of the cell cycle. And what cyclins are actually going to do is they're going to activate cyclin-dependent kinases. So cyclins and cyclin-dependent kinases are going to be actually working together. So you can see the cyclin here, which is a regulatory protein, is going to come into contact with a cyclin-dependent kinase. And one thing to take note of is there's a whole bunch of different cyclins. There's cyclin A, B, C, D, E, etc. And same goes for cyclin-dependent kinases. There's quite a few of them. So one of these cyclins will come into contact with one of the cyclin-dependent kinases, and what's actually going to happen is the cyclin is going to control the cell cycle by phosphorylation. And so when this cyclin and this cyclin-dependent kinase come together and form a complex, that complex is going to be binding to a target protein and then modifying it via phosphorylation. So phosphorylation is a process that we learned about. I don't think we put a name to it, but it's a process that we learned about when we were discussing the mitochondria and chloroplasts in plant cells undergoing photosynthesis, the light and dark reactions. And what we found is that ADP turns into ATP, right? So that is an example of phosphorylation. And here you can see that we have a phosphate that is now binded to the CDK. And we can call this CDK activated now. And this cyclin CDK complex is going to seek out a target protein. Now this protein that it targets, it's going to trigger some specific event in the cell cycle. Like for example, in interphase, you might go through some centrosome duplications, preparing for mitosis. So this cyclin and CDK complex that now is active and it has that extra phosphate group is going to target an inactive protein sitting in the cell and activate it by phosphorylating it. It's going to transfer that phosphate to the protein. This active protein is now going to go on in the cell and trigger that special cell cycle event that its purpose is to initiate. Okay, what happens next? Well, after that protein does what it's supposed to do, after the event has occurred, that cyclin is going to be degraded and the CDK is now inactive. Why is the CDK inactive? Well, it no longer has a phosphate, right? So it is inactive, and here's the cyclin. It is now degraded. It's been destroyed. When that cell goes through another mitotic division, preparing for another mitotic division in interphase again, remember, because it's the cell cycle, it repeats, more cyclin will be created when it's needed. So here we go. You have a bunch of different cyclins that are going to bind to, so you have different cyclins that specifically bind to and activate different classes of cyclin-dependent kinases. And I kind of brought this up in the last slide. And the only thing that is different here is you can see the cyclin concentration here. As the cyclin concentration peaks, as it reaches its peak, that is going to be concurrent. It's going to be at the same time as when their target protein is required in the cell cycle. So let's say we're at the point of the cell cycle where we need to be making more centrosomes. Well, at that point in time, the cyclin that is going to activate the protein that does that cyclin's concentration is going to be at its peak. And at all other levels of the cell cycle, that concentration is going to be, it's going to be low. So here, we don't need that to happen anymore, right? We do not need to be activating those target proteins. That would be a little premature, and we don't want that to happen. So at all other times, that concentration stays low. And you can look in this little image on the right and see that there's different cyclins. Here's cyclin E that's going to bind to the cyclin-dependent kinase type 2. And this happens in between the first growth phase and the synthesis phase of your interphase. And between the synthesis phase and the growth phase 2, cyclin A is going to be interacting with cyclin-dependent kinase 2 and cause some other event to happen. Now, this is not important. It is important for you to realize that there's different cyclins and there's different cyclin-dependent kinases, and they interact in different ways to cause different events to happen. But the specifics of which cyclin binds with which cyclin-dependent kinase when and what it does, that's something that you can learn about in university if you take biology there. And that's all we're going to say about that. So let's move on to the skill that we're doing today. The skill that we're going to be focusing on today is the ability to determine a mitotic index from a micrograph. So what a mitotic index is it's a measure of the proliferation status of a cell population. So in other words, it's how many cells are in the process of division as opposed to growth or, interphase. It's interphase versus mitosis. And this is going to tell us a little bit about what's happening in the organism at that time. So a mitotic index may be elevated during times where we need lots of division. You also use this, it's an important prognostic tool for predicting cancer or the response of therapy to cancer cells, right? Because cancer is basically cells that are rapidly dividing and are unchecked, and they're usually like mutated cells too, right? So being able to determine the mitotic index is important because we'll know if chemotherapy is working if the mitotic index decreases, right? If we're able to slow down the proliferation of those cells. So the first thing we got to be able to do is identify when is a cell actually dividing. So when is it in prophase, when is it in metaphase, when is it in anaphase, and when is it in telophase. And we want to be able to look at a micrograph and be able to see these things, okay? So here's interphase. Interphase, you will be able to tell that something's an interphase when there is one nuclear region filled with chromatin, okay? One concrete nuclear region filled with chromatin. Prophase, you know that a cell has begun to enter prophase when that nuclear region and the chromatin inside of it has started to condense, to supercoil into visible chromosomes. So here we can start to see visible chromosomes being formed inside that nuclear region. The nuclear membrane has started to break down here. And so this is the beginning of prophase. And so anytime you see a cell kind of like this is prophase. Metaphase, the thing you're kind of looking for here is the sister chromatid. So we can see a very beautiful sister chromatid with that distinctive X shape here. We can see sister chromatids aligning down the middle of a cell. So that would be metaphase, M for middle. In anaphase, you see the sister chromatids have been separated, right? So the sister chromatids have been separated and they're being pulled away from the middle of the cell. You can actually see, you can see that they're being pulled apart to the different spheres. So anaphase, quite easy to tell, the chromosomes are being pulled apart to their different poles. Telophase, these chromosomes are on their opposite sides now, and you'll be able to see two nuclear regions, two clearly distinct nuclear regions starting to be formed here, and the chromosomes are now gone. Instead, it's being turned back into chromatin, and you might even be able to see, if it's an animal cell, you'll be able to see the cleavage furrow, and if it's a plant cell like this one is, you'll be able to see the cell plate beginning to form. Okay. So let's practice. First off, we want to be able to identify which cells are dividing. So this cell here appears to be dividing. What phase is it in? Let's go back for a second. Let's review our phases. They're being pulled apart. That would be anaphase. Okay, so it should be clear. Look at this image here, and then look at this cell, anaphase. This cell is also dividing. This looks like prophase, right? It's in the beginning stages. This cell is dividing. We have two nuclear regions. Which phase is that? That's telophase, two nuclear regions. So far we have seen everything except for metaphase. Let's try to find one in metaphase. Here's one in metaphase. You can see it's aligning down the middle. And then let's quickly try to identify other cells. It looks like it's in prophase, prophase again. Here's an anaphase, another prophase, prophase, telophase, anaphase. These all look normal to me. Metaphase, anaphase, anaphase, telophase / cytokinesis, anaphase, prophase. This is very early prophase. Everything else looks kind of normal here. This also looks very early prophase. Anaphase, anaphase. And I guess that's it. Okay, so once you've identified the cells that are dividing, so now you're going to count the cells with the chromosomes. So I've already done this in the next slide. So here we can see we've highlighted the cells that are undergoing division. There is 20 cells undergoing division. And the mitotic index, how you calculate it, is you look at the cells that are in division. So there's 20 cells in division, and you're going to divide that by the total number of cells. So how many cells are there in total? Well, there's 55 that are not in division and 20 that are. So 20 plus 55, this would be 75. So you have 20 divided by 75. And 20 divided by 75 is 0.267. So there you go. Our mitotic index is 0.267 or 26.7%. That's how you calculate the mitotic index. What we're going to do very quickly before we end the stream for the day is we are going to switch our view here. On Schoology here, we can see our unit organization of life. We're coming to the very end of it. And here we are, cell division, the first, the one we're currently working on, and you see must complete. And what you have to do. So you must view the PowerPoint that we're going through. You must look at the ebook that we're using and go through the additional notes. And then here, open up the learning map, which is the guided notes that go along with these videos that you're watching and the PowerPoints with some formative questions in there. And then here's the first lesson we did on mitosis. This onion root tip lab, we're not going to be doing because we're doing distance learning right now. So if school starts off before the end of June, we'll try to get back to doing as many labs as we can. But this, I've changed it to, instead of must submit, must view. So just look at this and look at what we would have done And that's really all we can do for now. Here's the activity, mitotic index activity. So you're going to open this, you're going to click on the mitotic index, which is going to have you make your own copy. And this is going to have you look at a prepared cell slide of an onion root tip that has cells undergoing different phases of mitosis. And if you need a reminder of the different phases, here it is. And you're just going to answer these questions. So you're going to try to figure out how many cells are in each of the phases. So you're going to have to do some labeling here. Which ones are in interphase, which ones are in prophase, metaphase, anaphase, and telophase, and how many cells do we have in total? Then you're going to find the percentages. What percent of all cells are in interphase? Is that 50% of all cells? Right? So you're going to do some calculations here and then answer these two analysis questions, including calculating your mitotic index. All right, so that's it for the stream. Thank you very much, and see you tomorrow for our stream on cellular division when it goes awry. So cancer. See you tomorrow. Bye. Hey everybody, it's Mr. Emond. Hope you enjoyed that lesson. All my social medias are over here. You can like, follow, and subscribe. That way you get the notifications right away as soon as I post a new video. Up on the corner here, you'll find the playlist that this video is in. That way you can watch every video included in that unit, start it from the beginning, and learn how to do the topic that you're investigating. And as per usual, the next video in the series is right over here. Click it to continue your learning. Have a great day and keep learning, everybody. This is Mr. Emond. Bye-bye.